Okay, I hope the screen is visible.
Yes, ma'am.
Okay, fine. So, welcome you all to this sessions on oncology. So, first, let me introduce myself. I'm Pooja. So, I am been working with CliMed for more than 2 3 years right now. So basically, I have a, I'm working as a clinical researcher and also as a research scholar for my PhD degree. Also, like I almost finished my, PhD, and simultaneously I'm working as a publication writer and tutor here for CliMed. Other than that, I work as a research consultant for one of the clinical research organization, also as a medical reviewer for one of the digital marketing organization. So the oncology, so the very uh, the talk of the town, because the prevalence or the incidence of cancers are increasing day by day, right? And there are a lot of drugs coming into the market, so everything, so there are as you all, I expect you all from healthcare field, so there are a lot of fields that you can go to if you know the basis of oncology. Because either preclinical studies or in vitro studies, or pure research oriented study where you work on the cell lines or any kind of models. So the basics is important also, to know what kind of cancer, what are the therapeutics works, and what are the development or the research is happening in this field is very very important. So in this master certificate course on oncology, so we'll be studying on the in like the broad basics of cancers, okay, cancer, as well as we will study different kinds of cancers, okay, from sarcomas to GI cancers, breast cancers, so various kinds of cancers we are going to study in this whole certificate course. So okay, so this is about me. I hope you all are excited to start the session. So introduction to cell cycle. So this is where it happens. It is where because of that one particular cell in our body, uh, you know the tumor arises, right? Tumor doesn't pop up that way. So there is that one cell that induces or is a reason for the development of the tumor. So let's first study the basis of cell cycle. So what happens during the cell cycle? So when you know what is happening during the cell cycle, then when I talk about various mutations, okay, when I say there is blastoma and there is a RB gene mutation, or there is a BRCA gene mutations, and then when I say certain terms, it will be familiar to you all. So basics, I hope you all know this very clear, since majority of you are coming from the healthcare background. This is what we study since plus two, the cell cycle, cell division. But let us just brush up on this topic, so it makes us easier when we go to deeper towards learning oncology or the cancer biology, or the molecular basis of the cancer, or the various types of cancer. So okay, so why do cells divide? So the very simple question, why do cells divide? Why division happens? Basic growth, right? So
The
cells
growth.
divide. Yes, we are growing. So cells divide for 1 second. Just growth, repair, reproduction. So these are the major thing why the cell division happens. So basically the cell division, right? So what happens? It broadly divided into two phases. I think you guys, majority of you guys, very thorough with this fact, or know, these concepts are in your tip of the tongue. So these are nothing but interphase and mitosis and cytokinesis. So what is this interphase? Okay, so this is one of the longest phase in the cell divisions, okay, where the cell grows, where there is a DNA replication happens, and where there is any, also the cell gets prepared for division, because division happens in this phase, that is in the mitosis phase, right? So in the mitosis phase, like I told you, the division happens. So the cells, the nucleus and cytoplasm, they physically separate into two daughter cells, right? So there will be one cells, so that will divide into two daughter cells. So very basic. This is about the the major types. So if you have to see cell cycle, you can just focus on the cycle. So this is what I was telling about. So this is the interface. Okay, the interface, it will take up 80% of cell's life. So the interface starts with G1 phase. So G1 phase is a rapidly growing and it is a longest phase. Okay, so it will, based on the cells again. So certain cell might take up 8 hours, or sometimes it might take up years also to complete this G1 phase. So what happens during this G1 phase? So I told this is like preparing for cell cycle, or preparing this cell to become two daughter cells, right? So what happens, there will be more organelles forms, cells will have more organelles, so more organelles will be formed. And also they will start forming enzymes, okay, certain enzymes or certain proteins that is required for DNA replications. Okay, so most of the cells, if you're doing the cell cycle, which phase most of the cells exist in among these phase, then you can say it is G1 phase. So most of the cells exist in G1 phase, because this is the longest phase of cell division. Okay, so remember S phase. So there is a synthesis phase. So here what happens? Here the DNA replicates. So here the protein forms that is required for the replication. The enzymes forms, there is a requirement of polymerase, all these enzymes. So all these enzymes are formed here. And here what happens? There is DNA replication happens. We have two N cells. So that will replicate into 4N. Okay. Next, in the G2 phase, where what happens? Again, the organelles, they also replicate. The cell growth happens. There is an increased cytoplasma, level, and it will almost long last for 2 hours, and this is almost for 6 hours. Okay. So this is a interface which is consist of G1, S, and G2, where cell prepares to divide. So this is about the interface. So next, coming to the mitosis phase. Okay, the mitosis. So in the interface is done. So here where the G1, S, and G2 happens, then it comes to the prophase. Okay, so mitosis, we know, in the mitosis it will divide into two daughter cells. So why are we studying this? Why it is important to start from the very basic? So now in the in when I start seeing the treatment of certain cancers, for example, when I say when I start, okay, I will start with the pathogenesis of lung cancer, and then I say there will be certain targeted therapy, there will be certain therapy, then I give example of certain drugs, then I say wind blasting. So what is this wind blasting? It is a class of a drug. Okay, it is a drug. So what it does, it helps in the treatment of lung cancer, not just lung cancer, even for testicular cancer, so it is there in the market, or brain cancer as well. So what happens this, right? This wind blasting, it will inhibit it will inhibit microtubule function. Remember this drug. Okay, this will inhibit microtubule function. I hope everybody is following what I'm saying, and I'm audible, right?
Yes, ma'am.
Yes, fine. Okay. So this wind blasting, remember, it will inhibit microtubule functions. So if there is any error in mitosis, okay, so the cell division is halted. The cell division is halted. So no cell division happens. So either it goes to apoptosis, the cell will not divide. That will die there. Understood? Now So this is why we are studying the basics. Okay, so now you understood the interface where it goes to G1, S and G2, right? G1 every preparation happens, S phase synthesis replication happens, and G2 where every formation is now it is ready for cell division. So in the prophase very basic condensation happens. You might have studied in your in your higher primary school or in your 12th standard, condensation happens, the chromosome formation happens, microtubule formation happens, all that happens, right? Also in the metaphase what happens, they comes to this equatorial plate or metaphasic plate, it will come here, right, in the straight equatorial plate, right? So this microtubules I was telling, okay, so there is inhibition of this. Because of that what happens, there is no pulling in the NFS, this pulling will not happen. So there is, it detects certain kind of problems. Okay, see, there will be always a checkpoint. So this we will come later on, but there will be always checkpoint. So this microtubules are not working. So there is this the cell recognizes that there is something is not working fine, so that halts the cell division. Okay, so in the general what happens, so this might be working fine, but in cancer state we don't want the cell to progress, right? We don't want that tumor to grow, correct? We want that tumor to halt. How we can halt that? By avoiding this cell division happening, correct? So to render this progression, how we can render? Now you understood how it works. So this is how it works. So in the simple, in a cell division, what will happen? Prophase, metaphase, then it forms a two, then telophase, then cytokinesis happens. So in the cytokinesis what happens? The cell walls, there will be formation of the furrow. The furrow will be formed, the furrowing happens, and then that will divide into two daughter cells, right? So the cytokinesis, it is like I told, the final stage where the furrowing happens. Okay, so then they forms the two daughter cells. Okay, okay. So I hope you're clear with this thing. Basic cell cycle, interface, and mitosis. So any doubt so far? Anything here?
No, ma'am.
Okay, fine.
No, everything is clear, but can we get these slides later, man?
Sorry?
Can we get these slides later?
Yeah, definitely. We will be sharing this recordings later. Okay, so you will
Ma'am,
have.
even last class we had asked, ma'am, but they have not sent in the group, ma'am.
Okay, I'll make sure
Yes,
this
yes.
is shared. Okay, no
Okay,
worries.
ma'am.
Provide every slides, nothing
Fine.
that it will be beneficial for us to read after later.
Okay, fine. Please mute yourself so that I can start with the session. Fine. Okay. Next, we understood this process, right? Next, there is something I want to say about the significance of mitosis. So once a mitosis is very crucial step. Okay, so once a mitosis has happened, right? So if there is any changes in the DNA or if there is any mutations, once it started, all this process happened, then it is like permanent. Means if the two sisters are divided, means the two cells are formed, then it will constantly getting repeated. Means there is a formation of the mutated cells, you can say. So this is what happens. So it is very important to note that any changes that happens, that is recognized in the previous step. So that is one thing. The cell itself has certain checkpoints. Okay, they will be they will be having one. Okay, cell checkpoint. So now I told you. So the process, if there is any kinds of mutation or anything, so there are various checkpoints that will ensure the cellular conditions are, not gone further. Or if there is a mutation, they will halt the process in that way. So this is maintained by something called as cyclins. Okay, there are certain proteins called as cyclins. So they check this cyclin, it will not act, so it will not act independently. So this will usually act, or this will be activated by the protein called cyclin dependent kinase. Okay, cycling, this forms one a complex. So this will then act. So you can see during G1 phase, cyclin D is quite higher, and S phase cyclin E is higher, G2 cyclin A is higher, all this. So this, what it does is like a quality control. Okay, so quality assessment and all will be there. No, somebody writes a paper, so somebody checks the paper, whether there is a proofreading they will do, whether there is a mistake or not. If there is a mistake, they will send back. Or if you know, in a packaging also, if that package is not done properly, it will be rejected, right? The same way. So this cycling, this will check the quality, whether this can be gone further, whether they can undergo the cell cycle, whether they can form into two cells or not. If there is any if there is a mutation, of there is a certain kind of disruption, or any kind of problems in the cells, so they then halt it. Okay, so cell cycle checkpoints. Okay, so cell cycle checkpoint, basically, there are a lot of checkpoints, but the very common one is G1S checkpoint. Okay, G1S checkpoint, and there is G2M checkpoint, and then there is M checkpoint, or spindle checkpoints. Okay, so what it does is G1S checkpoint. So now you can see here in this section G1S. So between them there will be G1S checkpoint. Okay, so between G1 and S. So it is very, very important. So this divides whether the progression should go to S phase or not. So you know that S phase is where the DNA replication happens, where 2N becomes 4N. So this phase, this checkpoint, whether it will tell whether it should go to that point or not. Sometimes what it will do from the G1, if it feels this, the growth is not required, sometimes it will ask it to send to a G0 phase, which is a resting phase. Okay, so this is very crucial. So this checkpoint at which a cell chooses whether to proceed with the S phase and divide or not divide, or instead go to a the resting phase called as G0 phase. So this checkpoint is mediated by cyclin E. So you can see here S phase, G1 to S phase, cyclin E. Okay, particular kind of cyclin E, that is our cyclin dependent kinase 2 complex. So that complex will be formed, and two proteins called as retinoblastoma and E2F. Okay, so understand this checkpoint, right? So this is checked by this complex as well as these regulatory proteins. So now let's see what this retinoblastoma protein is. So this basically function is to prevent excessive cell division by inhibiting the cell cycle progressions. Okay, so when the appropriate signals for the cell divisions are present, RB, that is the retinoblastoma, is inactivated by phosphorylation. Okay, so I will just tell you, listen to this. RB is normally bound to a transcription factors, that is E2F. So once the RB is phosphorylated, okay, once the RB is phosphorylated, it will release E2. Okay, so this E2, what it does, it will bind to the promoters of the proteins required for cell divisions. So very simple, I will just do the diagram. So if you think this is RB, right? So it will always be, like I mentioned, see, RB is normally bound to transcription factor E2F. Okay, correct? So E2F, understood? One scenario. So this, let it be in the normal cell. What happens? Correct. So retinoblastoma, usually it will be bound to E2F. So once the RB is phosphorylated, it releases E2F. So once the RB, right, it is phosphorylated, the phosphate group comes and add to it. So this is a process of cell cycles. So it leaves. Okay, it releases E2F. It releases E2F. Okay, so what this E2F does? This E2F is able to bind to the promoters of the protein that are required for cell divisions. So this what it does, it binds to the promoter. Okay, so that will lead to cell division. Understood? So this will bind and the cell division happens. It will activate the genes that is required for the cell divisions. Understood? So in the case of, this is in the normal case how it happened, but usually it will be binded together, because of that cell division doesn't happen. So in the cancer cell, right, what happens, there is a mutation, right, in the RB gene or retinoblastoma gene. So this retinoblastoma, there is mutations. When mutation happens, there will be changes in the structural component, anything. Because of that, the E2F cannot bind. So what happens when there is a free E2F? What this E2F does, it will go bind to the promoter and it will do the cell division. So what happens, there will be overactivity of this E2F. There will be hyperactivity, or so there will be continuous cell division. So the cell division, what happens, continuous cell division leads to neoplasm. And finally it will lead to the development of tumor. Understood? But here it is in check. It will be gone when only it is phosphorylated, when the cell asks it to do. But in the case of mutation, only then this condition happens. So this is, it is commonly seen in retinoblastoma, the cancers, the tumor in the retinal cells, okay, retina. So usually seen in the children's, in the kids. Okay, so this is one of the conditions. So this is one of the knowing the cycles, how it works. So usually the in the chromosome 13 where this RB genes are present, so this is getting mutated and then the tumor is formed in the retinal cells. Okay, so this is why checkpoints are important. So the checkpoint defect leads to the formation of tumors. Okay, clear?
Yes,
Yes,
ma'am.
ma'am.
Okay, fine.
Yes, ma'am, it's clear.
Okay. So why is a cell cycle important in the cancer? Now you know why it is important. Because cancer cells lose this control of cell cycles. So they divide uncontrollably. So this uncontrollably leads to the formation of tumors. Okay. Next, so let's see what is wrong with this cancer cell. So why it is dividing that way? Okay, so now you understood this cell cycle way. So now let's see what is happening here. Okay, so in the normal cells you can see the diagrammatic representation here with plus growth factors. If there is a growth factors, there are certain cells. Okay, so there is a growth factors. So what happens, the cells start growing, and without the growth factor you can see the cell is not growing. But however, in cancer cell, with or without the growth factor, the cells keep on dividing. This is because the growth factor independence. Okay, this cancer cells, it doesn't require the growth factors, so it will work on its own. Okay, so they doesn't require any external growth factors like the normal cells. Also, what it does, the growth factor production. So what happens, this cancer cells, it might produce their own growth factors. Okay, so it will not depend upon the external source for the growth factors. Okay, because there are a lot of changes happening. Maybe the angiogenesis happening, the even the metabolism cycle is affected, right? So it will produce its own growth factors. Okay, so growth factor pathways issues also will happen because it will start producing its own uh, you know growth factors. That is depiction tactics, means it will manipulate the neighboring cells to produce more growth factors, and then they will consume the growth factors from that and they start growing. And then one more thing, factors called as contact inhibitions. So this is one of the hallmark of cancer. So what is this contact inhibition is, Very simple. When this you consider this as a cell, right? So it starts growing, so it start multiplying, right? So it will start multiplying. So 2, 2 to 4, 4 to 6, everything multiplication happens. But what happens in the general case, right? So this cells, they recognize, okay, now this growth had happened, okay, anymore I can't grow, there is some neighboring cells, so it will contact and then it will inhibit. Okay, it will contact the neighbors. Okay, now I don't have a space to grow, now I shouldn't grow, I have to stop this, growth or
Ma'am, can
this.
you please repeat the thing, the contact inhibition one more time?
Okay, contact inhibition, right?
Yeah.
Okay, so listen. When the cell starts growing, right? So it starts growing, it starts multiplying. In a general, in the normal cells, it will, generally what happens is it starts growing. So once it starts growing in this particular space, if you think, there are certain neighboring cells, right? So what it will do, it will contact this normal, the neighboring cells, and it will, okay, I can't grow anymore. There is no space for growth. Okay, so it will inhibit that cycle, or there will be no growth happening or no multiplication happening. So this will be simple, you can see the single layer of cells formation. But in cancer, what happens is this is not happening, the contact inhibition. So the cells simply divide. Okay, even if there are a lot of cells, okay, what it do, it does, it just multiplies. Okay, it will be multiplying upside here, there, everywhere. So because of that, again, that is the reason for tumor formations. So cancer, right? You can't just say cancer happens because of this particular cycle. Cancer happens because of lot of things. Okay, there are many pathways. Okay, so this is also one of the reasons. Means this is a properties basically. So cancer cells shows this contact inhibition. So they don't have that property. So they just pile up on. They just grow. Here, not like that. Normal cells, when they come to contact the neighboring cells, and if there is no chance for the space, or if there is no required growth or multiplication, they just stop, the growth is halted. But here in cancer cell, it doesn't work that way. So they just you know uh, keep on growing. So one more factor is that, so if there is any damages, right, in the normal cells, if there is any DNA damages happen, so usually cell, what it does, it will undergo apoptosis. Okay, but in cancer cells, it's not like that. Again, it starts growing. Okay, so this is one of why. So this is what is wrong with the cancer cells. Okay, this is what makes it strong. This is how it will, start forming the tumors in the cells. Okay, so along with that, so this is two things. What I wanted to differentiate between the normal cells and the cancer cells. So along with that, there is a activations, okay, of a certain enzyme called as telomerase. Telomerase enzyme. Okay, so let's see the beauty of this telomeres. Okay, so you know that if you have a chromosome, so these tips are called telomeres. Okay, so this is like a protective caps at the end of the chromosomes. Okay, this here, this is the telomeres. Okay, so what happens? Remember, with each cell division, this telomeres get shorter and shorter. Okay, so what happens? Uh, uh, as a person grows, his telomeres get shorter and shorter. Remember, this happens to everyone, and this is the process, okay, the biological process. So you can just say, as a person getting old, okay, your telomeres is getting shorter. Okay, so once the the telomere gets very, very, very shorter, so this is how it works. So in the normal cells, I told there will be shortening of telomere happening. Okay, there will be shortening of telomere happening. Okay, so once the cells, it will no longer has a capacity to shorten this telomere, so it will no longer divide. Okay, it will die eventually. Okay, so So in the normal cells, usually, okay, so this is a process. So the telomerase, there is an enzyme called as telomerase. So their activity in the normal cells will be low activity or completely not active at all or absent. Okay, but in cancer cells, okay, what happens? Okay, this will happen. Okay, the telomere shortening will happen, okay, but not as criticals, okay, because it is due to the telomerase activity. So in the cancer cells there will be increased activity, or they will be often active. This telomerase will be active. So what this enzyme will do, it helps to rebuild and maintain this telomerase length. So what happens now, I told certain level and then the dies, or it goes, but here it doesn't happen. It will keep on dividing. This, because of that, it will divide indefinitely. So the replicative immortality, okay, there will be divide, there will be uncontrolled growth happening. Okay, so this is also one of the factors, or one of the characteristics what you can see in the cancer cell. This is what I wanted to explain here as well. Okay, so now how the cancer develops. So very simple manner. So we have studied now what happens during mitosis. You understood the cell division happens, okay, wherein the DNA replication happens, cytokinesis happens, then there is a formation of the genetically identical cells, right? So in the cancer development usually happens in the multiple steps. So what happens? So there will be some damaged cells, right? Some initial mutation happens, okay. So what it does, sometimes it will die, okay, if it will be checked properly in the the checkpoints, everything. But sometimes it will try to avoid these checkpoints, or sometimes it will try to invade our immune system. So various mechanisms, again various pathophysiology. So again what it will do, again starts growing. So the cell division, and finally the second mutation, third mutations, and then it forms the malignant tumor. So it is basically the multi-step process. Okay. 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 Next, let's see the hallmarks of cancer. So there are basically 10 general hallmarks of cancer. So that is avoiding the immune destruction, tumor promoting inflammation, genomic instability and mutation, deregulating cellular genetics, energetics, oh, sorry, resisting cell death, inducing angiogenesis, enabling replicative immortality, activity of invasion and metastasis, evading growth suppressors, sustaining proliferating signals. So see, let's see one by one what happens. So sustaining proliferating signals, okay. This is what I just told, the contact inhibition thing. So sorry. So what happens, I told you, in the normal cells, if there is any kinds of issue, the cell uh, stops growing. So here it will not like that. They will sustain this proliferating signals, and it will just start growing. It just starts multiplying. So it will sustain this, the growth proliferating functions like that. Next, in insensitivity to growth inhibitors. So I told there are certain checkpoints, or there are growth inhibitory signals. So as well as growth factors which will promote growth, there are certain signals. So the cell says, okay, you are not supposed to grow now. Okay, so there are certain growth inhibitory signals as well. So cancers are unaffected by these external signals that inhibit the division of the cells. Okay, so normal cells exhibit cell dying or cell resting and cell dividing, but the cancer cells are not like that. Cancer cells, more cells dividing than dying, and cells may not go to G naught phase. Okay, so there are basically, there are three types of cells, if I have to say. One is proliferative cells, okay, which go on dividing, okay. Labile cells also we can say, like, the cells of our skin, GI tract, or hematopoietic stem cells. It's a really continuous, division happens, proliferation happens continuously. Cells divide, it goes to G1, S, G2, prophase, metaphase, anaphase, telophase. It's continuously growing. Okay, so this proliferative states. And then there is something called as stable cells. Okay, so like liver cells, like hematopoietic stem cells, sorry, the liver cells, the hepatocytes of the kidney tubules. Okay, so this what happens, there is a continuous division, but there is a limited, okay, there will be stable. For example, liver, okay, it will form the shape of, like, the how much liver is required, and then it stops, it's stable. However, if you even take the 40% of the liver, it will start growing again, no, they have a regenerative capacity, so they will come to the stable. So this is what is stable cells. And then there is something called as permanent cell, like, the skeletal cells, the neurons. Once if there is any defect to the neuron, it can't be treated because they're permanent cells. Usually what happens, they go to G naught phase, they will go to the resting phase, something like that. Because of that, there is no growth happening. So usually, why this is very important, these checkpoints, right? So sometimes what happens, either we can activate these checkpoints or inhibit this checkpoint. So drugs will be developed based on that, based on the receptors, based on the inhibitors. Okay, so drug, either they will inhibit certain process, or they will start or progress, or they will act as a receptors of certain process. So because of that, it will, the targeted therapy, which is a future, right? Nowadays treatment is not like randomly giving, the medicines and all. So it is like targeted therapy, right? So for one particular patient, they will see what kind of gene mutation is having, what kind of targets, what are the receptors. So based on that, the treatment is going on. So that is the future, because randomly giving a tablet, it doesn't work for all kind of cancers, basically in cancer treatment. Okay, next, invasion and metastasis, very common, right? So this is where the cancer starts spreading. So this is this is the last stage. During the last stage, where the treatment doesn't work, because the cancer has spread to the multiple organs, right? So metastasis. So you will study all this when it comes to the particular kind of cancer. But what is the process? Right? So metastasis, again, it happens. Either simply like that. So if if there is one particular tumor, means you can see this tumor, right? Okay, so either some breakthrough happens, okay, and this, the cells goes in the blood, and this will affect other parts of body. So you can see the primary cancers here in the colon. So it will goes and spread to the liver, it will go and spread to the lungs, that way. Okay, so this is one of the process. Or second, through lymphatic system. Okay, through the lymph nodes. So that it first, the lymph node gets affected, so the next lymph nodes, so because of that, and it will go to the next organs. Okay, so this is how metastasis happens. Okay, this is how it starts affecting, the various organs, or how the cancer spreads to the other parts of the body, either just like that. So that is why, right? So there is something called as the new technique. Okay, there is called as liquid biopsy. Okay, so biopsy, what biopsy is. So what happens if a person is having a liver cancer? So what they do, they take a tissues, the liver tissue, the hepatocytes, and then they see, okay, what kind of tissues, okay, all that happens, all the cells, whatever. So they understand, okay, this is a cancer. But the new development techniques called as liquid biopsy. So what happens is, so they take the blood, okay, and then they see for organoids, okay, because what happens, the blood will have the traces of cancer in much early stage. Okay, so this is one of the techniques. So that is still in the in vitro stage. There are a lot of labs even in India, in South India. There are a lot of labs working on this concept, the liquid biopsy, and there are few centers also which will detect, means they take your blood samples and detect for several kinds of cancers. So again, microRNAs, all these kinds of different techniques are there. Okay, so as you learn deeper, we will understand what kind of RNAs, what kind of organoids for what kind of cancer. So again, cancer spreads, you can see common sites of metastasis in case of bladder cancer, bone, liver, lungs, breast cancer, bone, brains. So it varies based on the cancers. Okay, so usually that is how they, the physicians, they look for the primary sites. Okay, if there is a ovary cancer, they look for other sites as well. Liver cancer, so they might see some tumor markers in the liver. So that is how, because they have to see whether it is metastasis has happened or not to do the treatment. Okay, next. So this is what I was saying about the telomerase. So this is also one of the hallmark of cancer. So I told there is a shortening of the telomerase. So telomerase, when I say, this is like a course. Okay, again, the DNAs. Okay, so you can see how it happens. See, the shortening is happened, right? So the cell divides after 40 to 60 times, there is shortening happens. So this is in the normal cells. But what happens in the cancer cells? So this is because of telomerase, the numbers are same. There is no telomerase shortening happens. Okay. So this is about telomerase shortening. Next, evasion of apoptosis. So there is something called as apoptosis. What is apoptosis? So that is nothing but programmed cell death. Okay, when there is excessive DNA damage, okay, or any other abnormalities, okay, then apoptosis is induced in the normal cells, but it is not induced in the cancer cells. Okay, so the cells, if there is any kind of damage in the cells, right, so it will recognize and it will kill itself. Okay, so that is a programmed cell death. So in TP53, okay, the tumor suppressor cells, right? So if there is a loss of this tumor suppressor functions, so what it does, it will eliminate this critical damage sensors from the apoptosis inducing circuit. So this function is destroyed. So because of that, what happens? Again, there is a continuous cell division and the formation of tumor. Okay, very simple way. If there is a DNA damage, the external signals, the cell death. Okay, in case of cancer, P53, it will escape that cell death. Okay, next is deregulated metabolic pathways. Okay, cancer cells use abnormal metabolism to satisfy high demand of energy and nutrients. The normal cells, if I write like this, the cancers will be piled up, right? There are a lot of cells. So there is a high demand of energy and nutrients. So what happens in the normal cells? In the normal cells, in the presence of oxygen, normal cells burns glucose in a highly efficient manner. So how much ATPs are formed? 32 ATPs. But here it's not like that. Despite of the presence of oxygen, cancer cells decide of burning same molecule of glucose, okay, in an inefficient manner, that is anaerobic glycolysis. Okay, so what it will form? Just 2 ATPs, okay. So what happens because of that? So there's less amount of energy formations, right? So what this cancer cell does is it will start taking a lot of utilizing a lot of glucose again to form, because the energy formed is less, so it will automatically start taking lot of glucose cells, lot of things, and it will undergo the abnormal metabolisms. So why would the cancer cells seemingly unnecessarily choose this inefficient way to burn glucose? So this is a good process, but why this has the cancer cell choose this way? Because they need the rapid energy production. So aerobic glycolysis produces energy faster, which is crucial for rapid growth and proliferations. Also, very important, biosynthesis. Okay, so what happens? So during this, cell divisions, there are some byproducts formed. So that will help in the building blocks of new cells, also for faster formation or faster multiplications. So the next cells has to be formed. So because of that, there's a constant formation of metabolism, constant divisions to form, basically for the multiplications. So next is evasion of immune system. See, one thing is, our immune system is very strong, so it will usually detect any kinds of changes that is happening inside the system. So there will be T cells, there will be B cells. So all this, there will be natural killer cells. So what it does, it will attack whatever changes is happening. But this cancer cells, since it is happening from the body, inside the body, right, your own cells are you know um, undergoing certain kind of changes, right? Not something foreign comes, right? So what it does, it is very smart. This cancer cell will be very smart. So it will dysregulate certain kinds of immune system, our own immune system. Okay, so it will produce certain molecules that will suppress the immune system, or it will express certain kinds of protein, okay, that will bind these receptors, that will activate the immune systems, or it will produce certain kinds of and stop MHCs, they say that. So there are certain kinds of MHCs. So this, what it does, it will reduce this MHC activities. Okay, so because of that, what happens, the immune system doesn't recognize that, so it will go weak, and then there will be increased formation of or the multiplication of these cancer cells. Okay, so this is one thing, how it will evade the immune systems. But this immune system, when it evades, right, so this immune system can also act as a point where we can give the treatment also. So there is a immune checkpoints, right? Certain immune checkpoints. For example, if in a simple way, if I have to say, the immune cells, right? So either it is masked, okay, or masked by the cancer cells. The two ways, I told you, either it will diminish or it will produce certain kind of proteins or certain like that. So what it does, this immune has certain immune checkpoints. Okay, but this immune checkpoint, this will fail to recognize the cancer, because it is there what is inside the system, right? And it will tell the immune cells, this immune cells, okay, it is our own cells, let us not fight, let us not fight. Okay, but when certain drugs are designed, okay, so what these drugs do is, there are certain drugs that is acts as antagonistic, okay, for this. So this will, what it will do, this will inhibit, okay, this will inhibit this checkpoint. So what will do, this immune cell directly attack the cancer cells. Or else, in see, simple way, if I have to say, immune cells, okay, it will have to go to this immune checkpoint. So it's like, this is a labor and this is a boss. So it will always have to ask the boss, okay, should I kill, or should I do something to this? Okay, he's a traitor, he's a traitor. But this checkpoint, oh no, it will recognize itself as like, no, it's not what happens, our own cells. Okay, because this has sent some kinds of protein, it has marked the immune rebel, all that. But development of certain inhibitors, okay, of this checkpoint, what it will do, directly start attacking the cancer cells. This is how immunotherapy works. Okay, so then what happens, our own immune starts killing this, we start recognizing the cancer cells and start attacking. Okay, so this is how it will evade. So what happens, this is the how evasion happens. Okay, so healthy cells will be there. Okay, when it transfers to the malignant cells based of various functions, what happens, are all immune cells comes, okay? B cells, natural killer T cells, CD4, CD plus, and it will so start killing. So tumor suppression, okay, gene. When there is mutation in tumor suppression gene, this will be avoided, okay? Then it will escape, okay? Sometimes escape, and because of that, there will be growth of the tumor, and it will avoid also. So another factor is chromosomal instability. So this is also one of the hallmark. So what it does, this instability is associated with poor prognosis, metastasis, and therapeutic resistance. What therapeutic resistance in the sense? So usually, okay, breast cancer. So they develop a drug, but cancer is very smart. There is some property with the cancer called as chromosomal instability. So each time, sometimes they start changing. Okay, either there will be chromosomal abnormality, either there is certain another kind of mutation happens. So you design certain drugs, but now it is not working because there is chromosomal instability. Okay, there is some kind of structural or numerical chromosomal, you know, instability happens. Okay, either there is a gain of chromosome, loss of chromosome, or deletion, amplification, any kinds of changes. So because of that, it can do the immune evasion or therapeutic resistance, means what you design for that doesn't work. Okay, so this is what is uh, on the side of the chromosomal instability. Next is inflammation. So inflammation is also one of the hallmark of, you know, cancer. So how do cancer cells cause inflammation? So basically, they release pro-inflammatory substances are called cytokines that will promote immune cells functions. Okay, so there will be increased formation of this, uh, the immunes, all these cytokine factors, all these tumor necrosis factors, or so increase this, which is not required. So that will lead to the development of cancers. Okay, sometimes increased is not good. So this is also one of the reasons where there the development of autoimmune diseases, right? So increased level is not good. So increased inflammation level is not good. So that will also lead to the formation of certain kind of cancer. Okay, there are a lot of cancers. So along with that, inflammation also leads to the activation of reactive oxygen species. So this also leads to the development of cancer. There are a lot of cancers that associated with chronic inflammations, like uh, colorectal cancer. So there is an inflammatory condition called as IBD, if you have heard, inflammatory bowel disease. So that Crohn's disease, and also what happens, there are reports that people who have Crohn's disease, after certain 20 years or 30 years, they might get converted to colorectal cancer. So in the same way, hepatitis B, HBV infection, HCV, all this causes chronic inflammation in the liver, finally leading to liver cancer. Okay, chronic inflammation and lung damage due to smoking again causes lung cancer. So basically, disruption in the cell cycle and cancer. What we have to know, mutation in the genes that regulate the cell cycle can lead to cancer. How DNA, this mutation can affect the DNA repair mechanism, cell cycle checkpoints, tumor suppressor gene, and oncogene. So in the next coming classes, we will study about this tumor suppressor gene and oncogenes. So this is a basic where we saw how cell divides, what happens, and why it is important, because we can target the cell cycles in the cancer therapy. Okay, so many cancer treatment target cell cycles. So chemotherapy, what chemotherapy is? It disrupt the DNA replication and mitosis, preventing the cancer cells to divide. But what happens during chemotherapy? All dividing cells are damaged. Radiation therapy also serves, but targeted therapy, it will only target the cancer cells. So this blocks specific molecules, okay, that is involved in the cell cycle progression. So this is a beauty of targeted therapy. Okay, so future of cell cycle based cancer therapy, more CDK inhibitors, okay, that will avoid a lot of cell division happening, cell cycle checkpoints, and certain combination therapies. Yes. So this is about the first session. So you guys can, this is open for questions. So you can ask any kinds of question if yes. What is the function of telomerase? See, all this polymerase, telomerase, so it helps in the growth or expansion of that molecule. So telomere, it will start growing, okay, or that will be stable. Means the telomerase shortening is avoided, okay. So yes, telomerase is like a shoelace clip, correct. Next, why metastasis is specific from organ to organ? So, why it is specific from organ to organ based on the, see, usually it happens to the nearest organ, okay? So, if it's, kidney cancer, then it will go to the adrenal gland because it is nearby. Or, same way, pancreas means it will first spread to the liver because all this present in the lymph. Also based on the, the lymphatics. The how the lymphatic drainages goes based on that. So basically, it will affect the the nearby organs, and then it will affect the the later on organs, or based on the you know the lymphatic system where there is where it leads to, like that, okay. Next question, is there any cancer of neurons? So yes, neurons are the nerves, so that these are the permanent cells, so this doesn't grow, right? It will do not divide. So if there is growth happening of these cells, okay, so then that leads to the neuron cancers. So certain kinds of gliomas, neuroblastomas, all these kinds of the nerve cell cancers. These are medulloblastomas, that is in the cerebellum, all that. Yes, you are right. Okay. Uh, usually happens that way, but it doesn't mean it inhibits sometimes. So it happens, the cell division happens with this RB and E12 complex, okay, but it is regulated, it is maintained, okay, how much cells need to be divided, only then there is a check for everything. But in case of cancer, this check is not occurred, so they just keep on dividing, okay. Next, can cancer cells be used as stem cells? Cancer cells, why? Like, I didn't get the questions. Like, like cancer cells are the stem cells or used in the kind, like what kind of use? I didn't get like that. Can you please briefly tell what this question exactly it is, or you can just rephrase me that question? Yes, can cell death can be induced. Various factors. Okay, chemotherapy, all this induces the death, right? Okay, so for the regeneration of organs, no, see, it will, either it will never develop into an organ, okay, so it will just form a tumors. So there will be abnormal cells that will divide into a tumor, okay. So there will be no proper differentiation happening like the stem cells. Stem cells are something that has a capacity to divide into a whole organ, right? It can be manipulated, and the hematopoietic are forms organs. So this totipotent C it has means it will divide into any kind of organ. So that is also still in the research. So, but a regeneration of organs, no, but there are certain studies that is happening. Okay, maybe if you give the particular growth factors, okay, or some kind of enzymes to grow that particular cells. So but it doesn't happen in the body. Maybe in the study we can go in vitro studies and all that. Yes, any other questions? See, simply to answer this question, you have to remember cancer cells are nothing but the group of cells. They are just uncontrolled divisions. They doesn't have a purpose there. Okay, it's not like dividing to form an organ like liver or heart or anything like that. So this just a mass of cells. Okay, and you just saw we saw the various characteristics. Okay, so they doesn't have they evade bodies immune system, and also the metabolic, you know the energetics, right? So they just utilize it, so they doesn't have a proper purpose. Also, the genetic instability what I just mentioned, there doesn't have a proper, the genes are not proper, they just undergo certain mutations. There will be chromosomal abnormalities. Because of that, what happens, there will be the behavior or the phenotype whatever is formed is not of good use. Okay, so this is a simple what I wanted to say about the regeneration of the organs. Maybe in the future they might find a study in the in vitro labs where they take certain, you never know, because studies are happening like that. They direct certain growth factors since had a capacity to grow. There might be certain study, there might be certain attempt to do that, but not in uh, the real time. It doesn't work that way. Yes, definitely you're gonna get the recorded session. Samriddhi, Samriddhi, are you there?
Yes, Dr. Pooja.
Yeah, please, we will share them this recording
Yes,
session
yes, yes.
as possible.
Yeah, sure.
Okay, sure. So I hope there's no further questions. I think we can wrap up with the session. Well then, thank you everyone for your time and patience.
Thank you, ma'am.
Thank you.